Deoxyribose nucleic acid, or better known as DNA for short, is a molecule responsible for all life as we know it. The most notable part about DNA are its complementary base pairs. These complementary base pairs are adenine, thymine, guanine, and cytosine are nitrogenous bases and such only bind to their respective counterparts via hydrogen bond. Adenine binds to thymine and guanine binds to cytosine. These four bases can be separated into two groups, purine bases and pyridine bases. In relation, it is double helix structure. It was also discovered that DNA is arranged in 5 to 3 and 3 to 5. All this means is that the nitrogenous bases are complementary and parallel but move in an opposite direction. The reason for this is that 3-OH can only synthesize in one direction. The leading and lagging strands in DNA are in relation to the replication fork, which is a structure formed by helicases. Essentially, the replication fork is just an area where helicases remove the hydrogen bonds of a strand of DNA to replace the strand. The leading strand of DNA is the strand that moves in the continuous direction, whereas the lagging strand moves in the opposite of the replication fork. The molecule DNA gyrase works very closely with the DNA replication and prevents many complications such as negative supercoils advancing the replication fork. Gyrase is imperative. RNA plasmids are strands of RNA that are synthesized along the first strand of DNA. It is the information of RNA plasmids that eventually results in the synthesis of primers. Just like RNA, DNA has similar molecules that catalyzes DNA synthesis. These molecules are known as DNA polymerase, and they play a vital role in creating brand new DNA strands. In the process of creating entirely new DNA strands, SSB or single strand binding protein has a very important role. The molecule prevents two DNA strands from rejoining up upon separation by binding to a single strand of DNA. When DNA helicase removes the nitrogenous bases of DNA, sequences of DNA known as Owasaki fragments are known to synthesize sequences of DNA separately. These sequences are then placed into DNA strand by the molecule DNA ligase, which is in charge of DNA repair and rejoining. In cells that contain a nucleus organelle, telomeres are sequences of DNA repeating over and over again. Similar to the end of a shoelace, telomeres protect the ends of DNA from damage and wear. Without telomeres, DNA would become too damaged and cells would cease to function. The origin of replication is a particular sequence where replication starts. Replication of DNA ensures that every daughter cell attains the appropriate number of chromosomes. Since DNA always retains at least one original strand when undergoing replication and doesn't create two new strands, it is known as semi-conservative substance. The end.